Fibrosis is a major health concern because it's associated with inappropriate formation of scar tissue.
And it has been estimated that fibrosis is responsible for up to 45% of all deaths in the developed world.
So normally when there's injury or inflammation, you have a process of inflammation followed by resolution of that inflammation. And then you have fibrosis in a physiological context to try and repair that original injury or inflammation and replace the inflamed tissue with more or less the same components. But actually what happens in fibrosis is that inappropriate matrix is made and scar tissue formation occurs. And it's not exactly the same as the original tissue. It has different mechanical properties and it has different molecules within it. And it's increasingly common in all the vital organs, in the heart, in the liver, the lungs, and the kidneys. And it's associated with obesity, which is a major global burden with increasing prevalence.
In obesity, tissue fibrosis is often undetected, it is difficult to reverse, and it interferes with the treatment options for obesity and its related metabolic complications. The rising rate of obesity worldwide has made this link between obesity and fibrosis an increasingly important public health care issue, and public health care initiatives focusing on reducing obesity might also mitigate the growing burden of fibrotic diseases. Our lab focuses on understanding the role of mitochondria in the pathogenesis of obesity and fibrosis. Our central research idea is that mitochondria abnormalities play a crucial role in the development of metabolic disturbances and fibrosis-induced functional adipose tissue in obesity. And by exploring the molecular mechanisms that link mitochondria dysfunction, fibrosis, and obesity, we aim to identify novel drug candidates and develop new treatment strategies to address these two interconnected health challenges, obesity and fibrosis. We have already established a wide adipocyte spheroid model which enables precise measurement of mitochondria variables under various conditions. But next step is to mimic adipocyte fibrosis and to create co-cultures of human adipocytes and immune cells in chronic inflammatory microenvironment. And we envision that our new 3D culture model of human-wide adipocyte spheroids will allow us and also others to investigate molecular mechanisms driving mitochondrial dysfunction and fibrosis, and also to assess novel drug candidates in physiologically relevant conditions.
So we know that obesity places a significantly elevated mechanical burden on all the joints, the knees, the hips, and the spine as well. And for many years, we thought that was just additional weight. But it's clear now that obesity and adiposity contribute biologically to the inflammation and catabolic activity within the joint. So our research is to try and understand the mechanistic links between obesity and fibrosis. So in order to be able to do that, we need good models, and we need to better establish in vitro models to mimic that. Currently in the lab, we work with articular chondrocytes and with cartilage explants from animals, humans, as well as a number of cell lines that mimic the biological properties of macrophages, which are important inflammatory cells within the joint. So the scientific literature and current research is showing that macrophages undergo a phenotypic transformation, so they become more inflammatory in joint disease. And we suspect that some of this inflammation is linked to some of the obesity-related factors, adipokines, cytokines. So we want to try and mimic those in vitro, and we want to be able to block them. So one of the purposes of drug repurposing is to take drugs from another disease, test it in a disease where it hasn't been used before to see if it can counteract the effects of that fibrosis. Now, is it going to be a treatment for osteoarthritis? Not really, because the real treatment for osteoarthritis is, in the context of obesity, is weight loss. But then once that has happened, then you need to be able to sustain the weight loss, and you need to be able to provide the drugs and the interventions that can maintain joint health. So we're hoping to find a number of drugs that can support joint health and joint function and not only counter the effects of inflammation, obesity-related inflammation, but prevent the fibrosis from happening within the joint structures.
I think the future of obesity treatment lies in approaches that target both mitochondria dysfunction and fibrosis. And by improving mitochondria function, we can restore the normal metabolic homeostasis in the body while reducing inflammation and fibrosis. This type of treatment approaches have the potential not only treat obesity, but also alleviate obesity-related metabolic complications such as type 2 diabetes and fatty liver disease.